Hello, everybody. Welcome to Microbiology Journal Club. I'm Fazal
Hi.
Lam, and this is my colleague and co-host Danny Chan. We're here to talk to
And
you about microbiology, about the phase 1/2 clinical trial of a COVID-19 RNA vaccine. Um,
yep. Yeah, this is a bi-weekly series, and on our off weeks we choose papers to read, and then on our on weeks like this week, we dive into the figures and we do a sort of a post-publication review. Or actually, in many cases, we read preprints these days, so uh,
yeah.
pre-publication review in that sense.
So, which is the case for this one, I believe. This is a medRxiv preprint. Um,
hmm
yeah, and like with a lot of the COVID studies, we're often seeing on like preprints first because everyone thinks that they're all very important for people to know about, I guess, because we are in this massive outbreak situation. Yeah,
Yeah. I mean, it's part of the speed of, sort of an increasing speed of scientific progress these days. I think that everyone knew that like the result science always happened right
mm,
behind like the official publication, but now there's like widespread acceptance of that, and everyone's racing to show us data.
yeah, and there are so many vaccine candidates out there. I mean, one of the big ones is Moderna, but we haven't had any papers from them about in humans yet, as far as I can tell.
Right.
But
And
yeah,
oh, I was gonna say, and Moderna is that RNA vaccine, which is why we chose this paper,
yeah,
because just to read a bit of data from a similar type of vaccine.
yeah, and so maybe a week or two ago, a study associated with Pfizer and another company came out with their like first tests of their RNA vaccine candidate, which
hmm
uh,
Is that other company BioNTech?
yeah, BioNTech. So I assume that
Uh-huh.
BioNTech created the vaccine because it's designated BNT
Right.
162B1. Um, so
It's a very classic innovation in the pharma space way of working, right? Like there's a small or medium-sized biotech that brings a product to a certain point, and then a large pharma company will either do some sort of split deal or acquisition, right,
yeah,
in order to bring that to like a later stage. Because clinical trials are extremely
mm,
expensive, and not every small company is going to have the resources to do that.
yeah. So
Um,
so
but the small company is going to hit the IP.
yeah, and the large companies like we've got a pot of money, a pot of gold. You can,
Yeah.
and
Yeah.
yeah, and so this is an interesting one. So like whenever I take a clinical trial, I do always try to like print off the clinical trials document just so I can. Well, not the this one isn't a
Is that
trials document. This is just the registry, so you can see.
is that from the clinicaltrials.gov
Yep,
website?
clinicaltrials.gov. So most
So that's the American version, but it's basically the go-to, I think.
yeah,
It's everyone else is looking to America to innovate these drugs and the regulation.
because America has loads of money, so they can actually do quite a lot of impressive stuff with their research.
I mean, I think that's a really funny thing because it's not like loads of money, but like relatively speaking, right? Because it's not the government that has this loads of money to do it. It's the companies that are based
Yeah,
here, right? And doing their trials. It's like if you want access to this large market, then you have to do the trials
yeah,
here. And like because that's being kind of the de facto standard, like there are other large markets now, right?
mm,
Like I mean, China is obviously a huge emerging market,
right,
right? But like because the states has been this way for a certain amount of time, right? Like I think people look to the states because a lot of companies' attentions are focused on accessing the market here,
yeah,
right? So the regulatory agency of this market ends up being like the de facto thing, even though it still has to be like validated by each other's like countries' markets as well,
mm,
or like regulatory bodies as well. Yeah.
yeah. So
Yeah. So this is phase one and phase
yeah,
two slash one slash two.
not phase half as I incorrectly said last time. Um,
Right. And this means humans,
yeah,
right? Once you enter the phase one and two, we're talking about humans.
right.
Not a lot of humans, though.
Yeah, so this is interesting because this is actually part of a much larger study. So the actual big study, they're like trying to power up for about 7,000 people. Sorry, let me see how many people. So they yeah. So this is they're anticipating the study will be complete in 2023, but the actual like numbers of people is going to be quite a lot larger. But in this case, this is
Yeah.
what we call a Sentinel cohort. So this is like the first 15 people, so that they can test out whether something check whether like the dose ratio could actually be like harmful to people, because if in case like your vaccine might have some nasty side effects, you want to do it start off in a very small cohort to see so that the less amount of people, so the minimal amount of people are affected by that. Um,
Yep.
so
Yeah, so I mean this is I mean that is the essence of this sort of phase 1, phase 2,
mm,
phase 3 like clinical trial progression, right? The idea is that phase 1 is supposed to be to figure out if it's safe,
yep,
right? Like will people like feel bad if they take this? And then phase 2 is designed to figure out how much can they take until you see some sort of effect. And then phase 3 is to be like, let's now take a huge the the statistically significant cohort,
yeah,
right, and see whether or not this thing is going to hit the predetermined efficacy endpoints as you discovered in phase 2.
yeah.
So the fact that they're doing a 1/2, right, sort of implies that they want to do them together.
Well, I mean, it's part of the compressed
And
timeline. They want to do everything very fast. So while they're
right.
doing the phase 1 trial, they're thinking, well, how about we take some blood samples to see whether it works or not at the same time, because
Yes,
that way
yes,
they're not
yes,
losing any time, and that there is a
yes.
what's the harm kind of aspect to it. Um,
Yeah, yeah. And I think the other thing, but like to your point, that the study is still enrolling,
yeah,
right?
it's still enrolling. So let me check through this because I highlighted it because yeah, it's still
Yeah.
enrolling. They're still like updating their their outcomes, and this is one of like four
So
can
what we're seeing, yeah, so what we're
yeah,
seeing here is just like is I mean they say it in the title, it's an interim report,
yeah,
right? We're not seeing the full phase 1/2 study being presented here.
yeah. I mean, the trial itself is looking at four different vaccine candidates. Of this study is only showing one, and the
Yeah.
trials.
Were you able to find like what those other candidates
I
were?
was not able to find them. I assume that they're
Me
proprietary.
neither.
Uh,
Yeah, I looked too. I was like, oh, I wonder what these other ones are. And
yeah,
then I'm like, oh, I can't find anything. So like
yeah.
they only tell us what one of them is here. But yeah, the study actually has four types. I assume what they are is I mean, we can talk about like what this one candidate is, but I assume they're all variations
Yeah.
in different ways.
I
Yeah.
assume they're all different parts of the spike protein, maybe, but I mean,
Oh no, I think it's different because
ooh,
like so I'll just say it, like the candidate they talk about like it's like a it's well it's a piece of RNA but it's not normal.
no,
They like substituted some of the bases they substituted a base for like a modified type of base.
yeah,
What did they call it, like pseudo uracil or
yes,
something like that?
that's because RNA.
Pseudo 1-methyl pseudouridine. I had never heard of that
Oh,
before. It was super
there's all
weird.
sorts of things because for RNA they get chewed up by the body very quickly. So in order to like make sure that they don't, they do try some base replacements for things that have the same effect but don't get like destroyed as quickly. So part of it's for
Yeah.
like making things up. So I remember a while ago there was like this thing about morpholinos, which are another type of pseudo RNA that did the
Mm.
same sort of thing. I assume this is another sort of version of that where it's like non-toxic, but basically it just means that your RNA will actually get there rather than because everywhere's full of RNases. RNases are in our air. They get shed from our skin. They RNA is just it's designed to be chewed up. So
Well, I've actually heard that that's a bias coming from like bench scientists, right? That like
well, I may
because
be
like I guess
biased.
in like because like in the lab like yeah there's RNases everywhere, but like for example like think about how they've been doing the testing, or I guess it could be fragments, but like this idea that like certain lengths of RNA maybe don't get chewed up easily because you can find them everywhere,
Mm,
right? Like when you do environmental sampling and you like swab different things, you end up getting tons of RNA. Right? Like if you're looking for and I mean that could be because it is encapsulated in an envelope or like it's inside of the cells, but yeah,
yeah,
RNA can be degraded
yeah. I
nonetheless.
guess it's a bench scientist thing of yeah, there's RNA everywhere, but never the one we want.
Yes,
Yes.
exactly. I think that's what it is. It's because like in bench science, like we're looking for very specific pieces of
Yeah.
RNA, right? That like maybe exist inside of cells, but not normally outside of
Yeah.
cells. And so like we get this idea that it's like, oh God, like that's so hard to do. Why did my experiment fail? It's those RNases that are everywhere.
Yeah. And what else was it? Oh yeah, they only the age group in here was 18 to 55. The full trial was 18 to 85, but obviously the reason I presume they decide to start with the younger group first for the safety trials is because the young group are kind of more like any side effects are going to be a lot less severe in the younger group, so you're not going to get any really bad stuff happening. So that's why I assume they only start.
So yeah, I mean, yeah, I agree with that hypothesis. I mean, we're seeing this, I think we spoke last time that like, it's uncommon to think that drug companies will publish like everything as
Yeah.
is, right? Like everything goes through like a lot of scrutiny before it gets published, and so they're not going to publish something that's going to look bad for their investors.
Yeah, which is why I worry when I hear a press release and go, okay, what's the study? I'm like, what study? And I'm like, oh
Yeah.
god.
Yeah. Until, and again, like it's not like they won't be allowed to move forward until they show right all of the data. I think that that's like
Yeah.
that should be reassuring for people to know that like it doesn't mean that like things are gonna get through because like the companies hide secrets, but it does speak to sort of scientific knowledge, right? And this is true in the publishing world as well. Like people don't really there's a bias in the literature for positive results.
Yes.
And it's like only emphasized even more when it comes to companies, because like they could just be like, we're not even going to deal with that anymore, right?
Yeah.
Because it's like really not worth the money to show people the mistakes.
Which is why like clinical trials are so great, because you can actually see what's in the file drawer. You can see what they're not showing you, which is why they're so.
Yes.
That's why I love them so much. That's why I always have that clinical trials document up on the side, so I can see like, oh, this is what they're not telling me, because it's usually not suspicious, but it's usually just kind of interesting to keep things in perspective.
Yeah, we say good things about open science a lot, right? And like the kind of open science ideals. And this idea of pre-registering your study is like very standard in the clinical trials world, because when you choose a statistical test, right, you're supposed to choose it before you do your experiment, right? It's not. If you want statistics to work for you, right, the whole idea is that like you design it and then you execute it, not you collect a bunch of things and then you figure out what's significant afterwards.
Yeah.
And of course, like science is messy, and so like there is that process of like doing some experiments and going back and forth. But at the level of clinical trials where we're dealing with humans and human data, it's been made very explicit by the regulatory bodies that they want to see these pre-registrations of studies. Yeah.
And I think it pays off a lot in terms of like efficiency in science.
Yeah. Yeah, I mean, at the end of the day, I think it's good because if we, there was a push at 1.2 to pre-register your like bench science experiments. I mean, maybe not every experiment, but like at least like the broad scope of like a study. Yeah.
I mean, that's still going on. I mean, one of the things I'm in different publishers, I've heard people trying to do pre-registered reports, and I think there is that kind of opportunity there to do that, and it would be really great if we had that sort of. But I think that mostly like it's just very difficult to implement, and no one really has figured out how. People want to do it, but it's just the figuring out the how.
Right.
But yeah.
Okay. Anyways, yeah. Sidebar.
Anyway, yeah. So this vaccine candidate was also quite interesting because they had like this Foldon domain, which essentially means that they trimerizes. So you get 3 versions of the protein creating a small microparticle. So effectively, it's forcing your body to make a version of the vaccine, which is quite interesting.
Yeah, like really bizarre because it's not. Again, they're not using the full spike. Well, how could they use the full spike in this case, right? Because it has a transmembrane domain, the full spike. But I actually really don't understand why, and I don't think it's explained very well why they make that trimerization domain, right? Like, so the receptor binding domain gets made. It's encoded by the mRNA. It's popped out, and then it aggregates with groups of it, and so now you get like a little cluster of weird receptor binding domains.
Well, yeah, I guess like the cell would interpret that as there's all this trash here that's not doing anything, and let's get inflamed.
Maybe the particle size might be bigger. That could be something. Yeah. It's weird to me. I mean, very strange. I tried to look in. I mean, the references didn't really help. I found to explain why did they use this RNA that was modified in this way? Why did they use this fold-on domain? Except in very broad strokes, right? So for the RNA modification, that PseudoRNA or whatever, they had published some like cell culture data saying that it actually expresses higher. And only in mammalian cells. So it's like some weird feature of like mammalian transcription systems or translation systems, that like for this thing works this way. Yeah, and the Foldon domain, I could only the thing that they reference in the paper, it just shows you like the biochemistry of making that trimer,
Yeah.
but not like the
trimer.
relevance of trimers in some sort of vaccine strategy.
Yeah, I had to do a fair bit of Google searching before I found like this paper on like creating vaccine nanoparticles, and this idea that creating like weird structures using like kind of these proteins could help boost. But even those articles were super vague. So I didn't get to the.
I mean, it could be the case of like it's like an emerging technology, right? So like all the papers are like they're kind of obscure and they're not exactly what you need, and so like reviews bring things together, but then it's not like you can't get to like a real satisfying description of what's going on.
And also, I think the science of adjuvants is there is a lot to it that is kind of a bit hand wavy at the moment. I mean,
yeah.
Also, like I know that for like some of my vaccine experiments, I'd just use BCG as an adjuvant, or like various other things used as adjuvants, like aluminium, and how do they work?
Yeah. So that term adjuvant, right?
Yeah.
This is definitely we'll probably did we talk about it in the other vaccines? I don't think we did.
I know.
Yeah, but basically people have been giving mysterious immunogenic other things with the target antigen in order to elicit the immune response.
I mean,
that's a good way to explain it, right?
Yeah,
it's like random immunogenic other stuff.
Yeah, so like this random stuff to make your immune system angry. And then like once you make the immune system angry, it points at this antigen and goes, that's what did it, and the immune system goes, and then the immune system decides to be angry or whatever antigen. I'm using very scientific language here, but
yeah, no, I mean, I get it. I got what you said. It makes sense. I think that is sort of the way to explain it. And you know, there is more technical language to go about it, but at the end of the day, like the explanation is never like very clear cut. Because there are many different types of adjuvants, and they're used in lots of different circumstances, often verified empirically.
Yeah, because the immune system itself is very complex, and how and why it makes decisions can be very difficult to parse out. So, at that point, like if something works empirically, you just go with that, because it might be several years before you figure out why, or because the immune system is almost complicated in terms of information storage as the brain. So it's, I mean, I had that in a lecture sometime a few years ago. So I'm choosing to think that as a fact, but
yeah, no, I mean it's certainly very complicated when you think about all the different cell-cell interactions that are happening. So the Foldon domain making these nanoparticles, so basically like larger than single receptor binding domains, clusters of these receptor binding domains. One could think of those as one could think of that function as somehow having an adjuvant function, right? Because the idea is that the antigen, it's not enough to just give the antigen, they had to give something else as well, right? And in this case they say, let's clump them, and that's going to be the antigen, or that's going to be the immunity boosting effect.
Yeah, anything that they'll make the immune system slightly anything that looks unusual that can like make the immune system like suspicious and investigate.
Yeah, actually I think that's a good point. Like often, I mean, one of the things that I've heard about adjuvants, the way people describe them, is that the adjuvants are to activate that innate side of the immune system, right? That like we want adaptive responses to this, right? But the adjuvant is like helping us transition to the right innate state. And so, right, I mean, I guess like this is going to get deep into some of the immunology, right? But two arms of the immune system, right? Like innate and adaptive. Adaptive is what's going to remember the infection and create the antibodies that are tailored to it. But innate is like always going on. Like, you know, there's tons of instances where your body just has to be like, this shouldn't be here. Yeah. And we need to remove it and respond to it appropriately.
Yeah, your innate response. They're like the beat cops and trash collectors who go around like cleaning up your body. And then you've got your Columbos and your like Sherlock Holmes who are like the adaptive immune system.
Yeah, I mean, there's like that anime series Cells at Work. Have you heard of it?
Oh my gosh.
Yeah, yeah, and I feel like they play with that they play with that analogy, and the metaphor breaks down in many
Oh yeah.
circumstances, but they try to take it as far as they can, like imagining like very specific anthropomorphized roles of the different cells and how they like work together as a
Yeah.
team in order to deal with an infection.
Yeah. Anyway.
Yeah. My hypothesis, my guess, my shot in the dark is that I bet they're trying different types of RNA. Like maybe it's different sections of it, but I also think they could be trying different
Hmm.
formulations of the either the LNPs or or the actual mRNA.
Yeah.
Right? Like maybe it's not always the foldon domain. Maybe it's other ways of
Yeah.
aggregating it.
It could.
Right? Maybe it's other RNA modifications and all that sort of
I mean,
stuff.
it could even be the lipid nanoparticle, or maybe it could just be like something that they do to make it patentable. I mean,
Oh gosh, it could be. That's true.
yeah,
That's kind of
that's if
horrific.
I'm
Yeah.
being really cynical, but yeah.
Right. Right. Another thing I want to say about lipid nanoparticles is uh, I, I wasn't able to find too many cases where mRNA is delivered into human beings.
Yeah.
The examples that I did find were these drugs. They deliver silencing RNA. So and yeah, and the idea is that like there's some rare diseases that are caused by like an increase of certain proteins. So like so you wrap up an RNA in there and you wrap it up in fats and give it into people and that RNA pairs up with the RNA that you don't want and they both get degraded. And in reading about lipid nanoparticles, yeah, it sort of started with drugs that were quickly degraded in serum, and they were like, oh, let's coat them in fats.
Yeah.
And then people found out that hey, those drugs like they end up getting into cells, and so maybe we could give RNA.
Yeah. I mean, I remember like a while ago, maybe like yeah, people working with lipid particles like in my lab and like using ultrasound to pop them in specific locations so that I mean, all sorts
Yeah.
of like crazy experiments you can do with lipid nanoparticles. They are actually wonderful tools, and they're great for drugs that you can't get in through normal means. Like, so I think
Right.
that they're a really interesting like because I think
Yeah.
vehicles are really important for like how you deliver the drug is really and especially like now they're developing smart vehicles where the I mean I'm sorry dragging away from the actual paper so
Yeah. Yeah. So I mean, no, but I think it's a good point to say to just emphasize that idea of we talked about Moderna,
mm,
right, which is an RNA vaccine, right, or like that
yeah
it's out there, we're curious about it, and it's also going to be delivered through lipid nanoparticles, but not all lipid nanoparticles are the
yeah
same, right?
yeah
Right? There's like so many different variables that they could be testing, and like in this case, right, like they describe us the formulation, and they're also doing four others, so there's a lot of different things it could be. And when we were talking about DNA, there was all those different ways of getting DNA into the cells, right? You could shock it, you could use a virus.
yes
That's the same with RNA as well.
yeah exactly
Yeah.
uh,
Okay. So yeah, so this is a safety study. So there, and it's interim, so it's not very big. They just sort of want to see is it going to hurt people. They do different
yep
doses, um, and that's part of like a safety slash a dose
oh yeah
escalation study.
yeah
Hmm.
they do the whole Goldilocks thing of like too much too little or just right um,
Right. Right.
yeah so I mean they start off with like 10 micrograms 30 and 100 which I mean I think the reason because 30 pops up a lot in like these sort of studies because it's like half in logarithms terms it's halfway, but it seems a bit because usually you think 50, but actually the way body reacts to certain things, looking at things logarithmically tends to uh, make a little bit more sense, I guess. Oh, in terms of like,
Hmm.
um, yeah, and
In terms of like it's like spanning orders of
yeah,
magnitude, right, to get effect, right? Because like what's the best way to span a range, right? And it would be like across all the orders of magnitude.
yeah, yeah, that's
Hmm.
exactly right. Yeah, and they look at the various side effects. So they've got figure one is just the traditional network diagram of like showing how patients are funneled through this process of the clinical
Yeah.
trial. Uh,
Yeah.
Which
Yeah. So randomized, randomized
yeah,
study, blinded it seems, so no one knows what's going on.
yeah,
They get some stuff and then they
yeah.
go.
I gotta say, I'm quite. I mean, there's so many open label studies that are going on. I'm quite happy to see a like a blinded study. It's like a breath of fresh air.
Hmm.
Yeah, and then like looking at the side effects, which is interesting because they do find that the 100 microgram like dose does start to produce side effects in at least one person. So again, these we're dealing with very small numbers. So like when you see something like 8.5% or something, that's just one person. Or
Yep.
so yeah,
Yeah, it's interesting. I mean, so I think an interesting point to bring up here is that if you look at like their, and I tried to allude to this before, if you look at their statistical analysis statement,
mm,
right? None of this is statistically significant because it's not
yeah,
powered to be statistically significant, right? This is not the stage of clinical trials where you're saying, I want to try it out a bunch of people and see what that effect is, right? Like, and extrapolate that information to like the population. In some ways, they're just trying to make sure, does can we give this drug to people or is it going to cause like big problems?
yeah.
Like no one's done this before. No one's put this compound into people. Like I even think they do a very bad job of pointing to the previous literature in this page,
Mm.
in this preprint, because I don't even know if they've used this particular formulation in animals before.
See, yeah, I don't think they have. I think they literally just jumped
it,
straight
it,
into
it,
people.
it it didn't seem like they did. I mean, they referenced like a flu study, right? But and I looked at the formulation of that piece of RNA, and it's different. It doesn't have the same modifications. It doesn't have that fold-on domain, right? So like, okay, there's a huge body of literature saying we've given lipid nanoparticles containing RNA, but like, how about this one in specific? And we don't get that. And again, there's reason to believe that it'll work,
Mm.
right? Based on all this previous stuff. And there's also a lot of reason to believe that it's safe because, again, lipid nanoparticles used in a lot of different
Yeah,
circumstances. RNA, really not that dangerous, right? When you're giving it to people, unless it creates something very strange. And so, yeah, they're giving it a shot in people to make sure that they can start expanding the study to then maybe look at efficacy. So they're not powered or even set up to look at efficacy at
yeah,
all, right? Pre the study, but they will because, as you said before, there's no harm in doing so,
yeah.
right? But from the standpoint of like, what is it suggesting to us, right, about the greater population? You have to take it with like a huge amount of
Oh yeah.
salt and like reservations, right? Because it's not for that. Like the the reason why they're doing it is just to make sure that they can try it again in more people.
Yeah, I mean, I'd expect this to be happening quite a lot, and the only reason that they've shown us this one is because the results seem fairly positive. Um,
Yeah, yeah. And in some ways they're teasing us, right? They're teasing us by showing us that preliminary efficacy stuff, right? But there's no statistical significance behind that
oh,
efficacy
yeah,
stuff.
no, this is basically a descriptive, like very much a taster. Oh, I mean,
Hmm.
it's
Hmm.
better than a press release because
Like
we
some.
can see the data, but then on the other hand, we have to be very careful because again, this is a very small, 15 people out of a study that's going to take out thousands. So I think like I mean, when experimenting in the lab, I always had the experience of when I start off a study, I do a preliminary version, and that shows something amazing, and then when I do a bigger version of it, it disappears.
Exactly. Yeah, yeah. And this is that mentality in the phase the three phases, right? Is that one is so that you can even put it in people, two is that preliminary study, and then three is the one where you're like, okay, now it's powered and we're gonna see what's going on.
Yeah,
So the fact that they're squishing one and two together, a lot of studies actually do it, even like outside of
mm,
this time of like rapid drug development. If you can make a good case for it, companies would like to squish them together because it's a lot less bureaucratic overhead,
yeah,
and they're probably granted the ability to squish it together in this case because they might have like an interim analysis planned in the study design, right? Like this is maybe part of that, right? Showing us this information is maybe part of the reason why it was allowed to do and allowed to go through in this way.
yeah,
Yeah. So injection site reactions, they're very typical. Those are very typical injection site
yeah,
reactions, right? You inject something weird into you, it's going to look like that. Um,
yeah,
just making sure that's a lot about I would think that that's a lot about the formulation, not really about like the mRNA at all. That's about like the way that they made those lipid nanoparticles.
yeah.
um, but then they do look at some systemic ones, and again, nothing seems too crazy. Grade 4, you know,
Yeah.
like basically anything can be really bad if it got to grade 4,
Yeah.
right? Like,
If you
yeah.
if you have to go to hospital, it's pretty bad. I think that happened like maybe once or twice, like where they like list like people. Oh no, yeah, they if you have to go to hospital, then maybe this vaccine is the best thing for this like outbreak. Um,
Yeah, so they do report severe,
yeah,
but they don't actually give us any grade 4s.
yeah,
So
they're just letting us know that they're looking out for them. Uh,
it's weird. It's weird that they use that language, mild, moderate, severe, grade 4, because like normally it's like 4 different
yeah, it's
grades.
usually mild, moderate, marked, and then severe or something like that. Usually, I mean,
Yeah, or something like that, or grade 1, grade 2, grade
yeah,
3, grade 4. I just don't understand the driving that.
mixing numbers and words. I mean, grade 4, where's your grade 3 and grade that sort of thing. I understand that. Like,
Yeah, I'm not sure. I mean, to me, it means that they're not using a scale. So, like, typically there's this like grade 1 to 4 scale that I see
hmm,
used, and like it's standardized. It just makes me think that it's not that scale, but I didn't look it up. It's like the
yeah,
CTACE. Oh
I
my goodness, I'm gonna Google
yeah. I mean, I'm just trying to
CTAAE.
look out for that.
AE is adverse events. That's like the word people use in pharmaceuticals to say side effects, right?
Yeah.
Or like safety issues.
Yeah, I mean these are mostly
Um,
self-reported reactions. So they gave people an e-diary, an electronic diary, to keep an idea of like effects or severe adverse effects. Um,
yeah.
but yeah, I mean, again, like, I mean, in terms of like self-monitoring, that's you're when you're running a vaccine study, I imagine you're going to get quite a lot of people with. I mean, if I have a hangover, it might be due to me drinking the night before. It could be the vaccine. So I mean, you'd want to like report both just in case. Um,
Right. Absolutely.
um,
And they don't show. Do they? They don't show the what's medication in this
yeah,
medication proportion.
medication.
Oh, that's people using different medications during this time.
Yeah, I think like to like medicate for any adverse effects. So if you're taking something, if you experience like a severe like fever or chill, whether you took medication, whether it's severe enough for you to actually go like, I need to take something for this. That's how I
Hmm.
interpreted it. I'm not sure whether that was the it was meant in the study.
3 were assigned to this placebo, and then 3 were assigned, but then they were all given dose 1.
Mm.
Right?
Yeah,
They got assigned, but then they were all given it. Is how this study works.
yeah. So yeah, I mean the thing is like for the 100 microgram group, they weren't all given the second dose because in the biggest dose group, they basically said actually a lot of people are getting adverse reactions. We don't want to give people another dose just in case it gets worse. So that
Right.
was like an interesting like thing that they did, and that also plays into how you interpret some of the later results. Um,
Yeah.
um,
Oh yeah. Sorry, I just found the term. It's Common Terminology Criteria for Adverse Events,
right.
CTCAE, and that usually is like yeah, it's like a uh, 4 grades, or actually it's 5 grades because there's grade 0. And like that's sort of like what everyone uses. So I like maybe they sort of use like a differential one at the beginning, and the grade 4 is like the
Yeah,
CTCAE version of it. That's my guess.
that makes sense. Um, yeah, and they list a bunch of like so they list also adverse reactions after the second dose as well. They also like took measurements of the lymphocytes in which I found quite interesting because after like the first dose they found like that the lymphocyte
Yeah,
count in the blood goes right down, which indicates that basically the lymphocytes are like kind of someone's blown the whistle and they're all gone on alert and gone into the tissues. So
yeah, that's true. Yeah, circulating lymphocytes may have gone into the tissues.
yeah, so again you're
That
yeah
Yeah, that's interesting. I mean, lymphocytes are also a common adverse event to look for,
yeah
right? Like sort of blood issues. But I'm really not familiar with like what adverse events look like in vaccines, right? Because like it's also very specific
mm,
to the types of drugs that you're looking for, right? Is like the types of sort of other tests that you might do to look for more subtle danger signs.
yeah. I mean yeah it does depend a lot on the virus or bacterium that you're working with. because sometimes you
Yeah.
get
Well, I mean, I think the formulation of the drug itself,
yeah
right? Because if the mechanism was known for this drug and it was like this drug like stimulates the immune system in a certain way, then like you might look for certain things, right, as evidence of what's going on. And then we get the teaser.
the teaser. Oh yes, the
Figure 4.
figure 4. Yeah, this is the classic. So 4A is looking at the reaction to the receptor binding domain, which is kind of the basic. You really want this to work because you're injecting them with the receptor binding domain. You at least want an immune response to it. So yeah, and they show that like the 30 microgram group has the biggest dose, but then you have to remember the 101 only got one like dose of the vaccine, whereas the 30 microgram got
Right.
two.
Got 2.
And they also compare it to convalescent serum. So they taking like people who recover from the disease and who have serum that will react to the virus.
Yeah, neutralizing. It's a neutralizing
Yeah.
test. They actually use the virus in this test.
Yeah.
Which I find quite fascinating.
Yeah, so that's in
I always love seeing that, right? Using the actual virus.
yeah. So that's in like figure 4B where
4B.
yeah, that's the really important test because we want this because an antibody can latch anywhere on the protein and you have to make sure it does something. And so this actually shows that it does something, which is really impressive.
And I hope that they end up doing that in the phase 2 powered one, or at least for some portion of the phase 2. It would be nice to see that data for all of them. Like, rarely will this type of data be used in phase 3, I think, because, well, let's look. Like, primary outcome measures. Oh no, that's not that. Secondary outcome measures, maybe. Yeah, spike protein binding antibody levels. I'm looking at the clinical trials website.
I printed it off
Look at the
because
things
I
that
was
they're doing.
highlighting stuff. And
Serum neutralizing antibody levels.
yeah,
Yeah, neutralizing titers through 2 years final. So right, like it's not the primary thing because the primary idea is to find safety and dosing, but secondary outcome measures will include this stuff as well. So it's nice. It's nice to see them set up, right? Like in some ways, this is also like the trial run for the study group, right? To make sure that they can make the data, they know how to analyze it, they can get feedback on that. Their investors can be excited that like there's positive results. That's why we're seeing this paper out.
Yeah, totally. If I was an investor, I saw this, I'd be like, because it looks good. But as always, we have to keep in mind that this is just a very small portion of a much larger study, and that things again, this might be a skewed population. There might be also other like effects that might be causing this because it is such a small study. Like someone might be just randomly very immune to coronavirus through no fault of their own. So you have to. So I mean.
Well, they have zero, but they have placebo.
Oh yeah, they do have a placebo group. Just
it's not like that's not what's driving it. I don't think.
But yeah, they have baseline as well. I think as well. So that also is.
Yeah, they have one.
So, but I mean, there are sorts of other like effects that can change. So I mean, there's this thing called the.
No, I actually think it's to me the reservations of interpreting this data would be it can't be extrapolated to a larger population.
Right. Yeah. That's the important bit because again, the population itself was 12 people, and from the makeup, let's see if I can pull out the baseline data because so I mean, is mostly white. There's there's mostly male. So it might not generally translate to a much larger population because you're trying to see the entire diversity of all the people you want to be treating in this one group. And so usually we'd power studies to have that in mind. But this one.
I think they're likely from a single center.
Yeah.
Like a single healthcare center. That has a big effect.
Definitely. I think like because I think you can tell which one it is because none of them are sent because they've got a long list of all these health centers recruiting, and none of them are recruiting at the moment apart from like I think 1, 2, 3. I think there's only a few of them who are actually recruiting for the study actively at the moment. But they'll spin up more as like.
Actually, that's really interesting. Like, what does it mean? Like, does the preliminary report have to be chosen beforehand? Like, what other data are they sitting on right now? Like, why was it these people that did a preliminary. I don't know. That's weird. There, there's lots of details, right? Because like it's not like this clinical study on clinicaltrials.gov talks about
Mm.
exactly this interim report,
Mm.
right? It's talking about the full study.
Yeah.
So like who decided this interim report was gonna be put out? It seems just like they wanted to give us a teaser.
Yeah, I mean, I assume that they're running like a bunch of like Sentinel studies like this concurrently, and this is the one that looked like the
Right.
the most impressive, or like at least that demonstrates their concept the best.
Yeah, yeah. So like inside of the pharma companies, they have these like huge documents about the actual
mm,
protocols, right? That go into very in depth how to do all the different things. But rarely are those going to be released until much later after something
oh
gets
yeah, those
published.
get past the regulatory authorities who go through them like with
Yep.
a fine tooth comb. But we
Exactly.
rarely see those
Exactly.
released to the public. Um,
Mm-hmm. Mm-hmm.
which I mean, that's an interesting discussion to have on its own because I mean, trying to because then these trial documents are really massive things, so you really have to take a long time to go through them. Um,
Mm-hmm. Yeah.
yeah, I mean,
And in some way it's actually really funny because like this is phase 1/2 study. This study consists of three stages: stage 1, stage 2, stage 3. So like even within this phase 1/2, they've broken up into three stages where they're going to do these sentinel studies, and then they're going to do it in larger groups, and then they're going to do it in the final candidates.
yeah, I
So
understand why they do that from the safety perspective because if something goes wrong and it starts showing up, you don't want to have it already in a bunch of people. You want to make sure that you ramp it up slowly. Just be careful, step by
yeah,
step. So like anything that's like
yeah.
really likely to happen will get picked up early on, and then as you get improve the size of your groups, you'll pick up the more rarer side effects, and then you'll be
Hmm.
able to like work out whether you want to cut it off before you start like giving it to a whole bunch of people. Um,
Right. And I think the other thing to remember is that a majority of drugs and therapies fail at the phase
yeah,
1 and phase 2 steps, right? Like that's the drug pipeline is crazy. Like, yeah, yeah. There's like a lot of work that goes into it, and a lot of it doesn't amount to anything. Which again is why drug companies work, the big drug companies work this way, that they partner with smaller companies to take on that risk. And only when they see some success, right, or the opportunity for it, do they shell out the money to start doing the studies.
yeah. And one of the amazing things we are seeing this in real time with the COVID outbreak. That all these studies they
Mm-hmm.
are rushing to tell us about their new vaccine candidates, and we are getting to see like each step of this and like where and there will be a few of these studies where they don't do very well at all, and
Yeah,
and that's going to be quite hard for
yeah,
everybody because everyone's hoping for rooting for these to be successful. Uh,
right, right.
so, which is why I'm always like trying to be very down on these studies because I just don't want to get my hopes up.
Yeah. I mean, it's reasonable to expect, right? Because like if you think about like what the actual success rate for new drugs or vaccines is, right? It's like it's low compared to, yeah, what's going on
Yeah.
here. I was just I don't know if I said this last time, but like uh I worked in Staph aureus, and like there have been many attempts through the ages to create a Staph aureus
Yes.
vaccine. And they've been unsuccessful. So, like it's, I think 20 or so, maybe more than 20 different attempts over the years.
Oh, yeah.
But yeah,
I mean,
the imperative here is greater, right? Because this is affecting a lot of people, and we sort of want one, so that we can imagine a world where we don't have to have so many restrictions on the way that we work and come together in groups.
and also to prove that we can do it. This is because we
Mm.
could get like any new virus to come out. It doesn't have to be coronavirus. It could be something else. But being able to prove that we can actually create and test a new drug and develop it very quickly is actually that in itself is the test that we are having
Yeah.
as
Sure, absolutely. I mean, like people talk about emerging infectious diseases, right, as something that not enough people are concerned about, right? That when SARS came through, right, we sort of knew how that happened and
yeah,
it caused this effect. Well, that happens all the
yeah,
time, right? That's the biology of our planet. And if we don't have systems and ways of working that allow us to rapidly create vaccines for those scenarios, right, and control those outbreaks, then we'll just find ourselves in this scenario again once everything settles down.
yeah. I mean, pandemics do happen a lot more often, and they're a lot more well known to happen these days. Especially with new strains of flu coming. There's Ebola that has
Higher densities of
yep,
humans. right, is one thing. Habitat loss for animals,
Yes.
right, and the motion of animals into human
Yeah.
populations. Yeah, there's like some very interesting like societal trends that actually drive an increase in pandemics.
Yeah, I mean, there's a lot of yes, a lot of interesting science going on in that sort of area. Something that the rate of discovery of like viruses and new thing just is going up because we're getting better at
Hmm.
detecting them, and our society is becoming so more internet. The amount of people I need to know in order to be in contact with someone who's in the rainforest. So in terms of that chain of transmission is shrinking as we get more
Yeah,
interconnected. So
yeah.
so yeah, if someone like
A more diverse genetic population, right? The increase of immunocompromised individuals in the society in our population.
yeah, in areas
Yeah.
of
There's lots of things.
yeah, areas of increased biodiversity, because then the more animals you get, the more microbes they're having, the more chances that one of those microbes will go into humans and turn us into zombies.
Hmm.
Uh,
Oh yeah, we're waiting for that cordyceps.
been training for
Zoo,
this.
yeah, zoonotic
I
cordyceps.
trained for the wrong apocalypse. Oh.
Okay, let's move on to the next paper that we had discussed, I think.
Yeah, okay. So say a quick bite, but for anyone watching on the YouTube channel, we are doing this two-part paper, so we're gonna be talking about dexamethasone next time. But for the moment, we'll say a quick goodbye, and we'll join you in a second.